Hi, thanks for listening. I'm presenting the non-parametric distribution clustering to find out the gene group behavior in the treatment targeting the glioblastoma. We believe that the method we propose can unravel the gene-specific profile associated with the treatment efficacy, and then we can find out the specific subpopulation of this gene group behavior, which can potentially lead to a better treatment. So, the data set we have is actually obtained from our collaborators' lab, and in their lab, they treat the mice with this glioblastoma brain tumor with different treatments, and then we obtain the brain tissue, go through the RNA sequencing, and then this is the data set that it looks like this with the two-dimensional matrix, numerical, and gene counts in each items. With this type of dataset, the most commonly used method would be subspace clustering. So there are many type of this kind of clustering. They all contain certain workflow with which is normalizing or and then dimensional reduction, and then some algorithm pick up the cluster them into different groups. But the drawback is couldn't it can't it can only link back to the certain spot of each cluster, but it cannot find out a gene specific um uh profile in this method. That's why we propose a distribution based clustering. In our proposed method, we assume each gene count follows a negative binomial distribution, and then hierarchically, we use the Dirichlet process that can force the data into a certain cluster. Um, this is the stick breaking process in the Dirichlet process method that can ultimately enforce the data into a certain cluster at the end. And then we also set up the mean and the dispersion parameter that following a gamma distribution. And then later we will implement them using a variational inference method, which can extend to very big data set. At the end, we will find out the average gene expression of the subgroup gene behavior cohort, and then the spread, which is their dispersion. Because our project is still ongoing, I can only show you the clustering, which is using the very traditional subspace clustering method. We can see the red area are the other tumor, and then this is the gene expression of a very specific gene, because they are selected because we find out they behave quite specially between different treatment. And yeah, this is it. We hope that our method can ultimately identify the gene-specific profile, which can successfully eradicate the glioblastoma brain tumor in the treatment. So then, and then it can lead to a better treatment and a higher survival rate for those patients. So, by understanding the molecular mechanism that regulator response to the therapy, we can hopefully develop more effective therapy that targets the underlying cause of the disease, and ultimately it can, of course, lead to improve outcome and a better quality of life for the patients with this brain tumor.